Hello and welcome to EHJ Today. My name is Stefano Taddei and I am an International Associate Editor of the European Heart Journal. We are speaking from Barcelona. Actually, there is the annual conference of the European Society of Cardiology. Today we are talking about a hot topic, which is the treatment of acute coronary syndrome, and more in specific the results of the SOLID-TIMI 52 trial. And my pleasure to welcome here Professor Michelle O'Donoghue from Boston, from the Brigham and Women's Hospital, and Professor Robert Califf from the Duke Medical Center from North Carolina. And first, Michelle, tell us more details about the rationale of the study and how it was conducted and the results.
Sure, so thank you. So the SOLID-TIMI 52 trial evaluated the efficacy and safety of Darapladib. So Darapladib is an inhibitor of an enzyme named LPPLA2. And that enzyme is believed to play a role in atherogenesis. So specifically, we know that that enzyme circulates bound to LDL cholesterol, gets delivered to atherosclerotic plaques, and the belief was that by hydrolyzing phospholipids, that it would promote inflammation within the plaque. And therefore, by inhibiting that enzyme, that we might be able to prevent further atherogenesis and prevent plaque destabilization. And supporting that concept, there were studies along the way, certainly several epidemiologic studies that showed that higher levels of LPPLA2 are associated with a higher risk of having a cardiovascular event. There are also some genetic studies, Mendelian randomization studies, that also suggested that LPPLA2 might play a causal role. So with that in mind, that Darapladib was developed as a therapeutic agent and then evaluated in phase 3.
So, and in the rationale you have some promising results from some analysis of previous studies. Can you tell why you decided to move to acute coronary syndrome?
Sure. So starting with the phase 2 program, Darapladib was evaluated using imaging technology, so specifically IVUS virtual histology technology. And they looked at different components of the atherosclerotic plaque both before and after treatment with Darapladib. And in the phase 2 program, the trial did not meet its primary endpoint, but there were changes in terms of progression of the necrotic core. And it appeared that Darapladib, compared to placebo, halted progression of the necrotic core on a background of statin therapy. So that was, one piece of perhaps encouraging news, although again, the trial did not meet its primary endpoint. And then Darapladib was evaluated in 2 large phase 3 trials. The first of which that was already presented was the STABILITY trial. And again, that trial did not meet its primary endpoint. That was an evaluation of Darapladib in patients with stable coronary heart disease. And although it did not meet its primary endpoint, there were some encouraging signals whereby 2 of the pre-specified secondary endpoints, major coronary events and total coronary events, Darapladib did significantly reduce those two secondary endpoints. So with that in mind, we now move to SOLID, evaluating Darapladib in the phase 3 setting, in the post-ACS setting.
Okay, so a brief description of the experimental trial. How many patients it was randomized versus placebo, and then the results.
Sure. So SOLID evaluated Darapladib compared to placebo in a double-blind randomized placebo-controlled design. It was 13,026 patients that were enrolled, and it was across a spectrum of ACS. So these were moderate to high-risk patients, unstable angina, non-ST elevation MI, and ST elevation MI. And the median duration of follow-up was 2.5 years, which was actually a little bit shorter than the STABILITY program, which had gone 3.7 years as its median duration. And the primary endpoint of the trial was coronary heart disease death, MI, or urgent coronary revascularization.
Okay, and the results?
So unfortunately, Darapladib did not reduce the incidence of cardiovascular events or major coronary events. So the effect overall for Darapladib was neutral as compared to placebo.
Okay. And Robert, just a comment on the disappointing results, I can say, because there was a great expectative and but no effect of the compound.
Of course, this is of course a hard thing about cardiovascular drug development is you just don't know. Even blood pressure as a surrogate now is under fire because we have treatments that reduce blood pressure that don't produce a benefit or may even produce harm. And we've just had a variety of drugs that affect LDL cholesterol turn out to be disasters. So you have to do the trial to find out, and this was done. It's a pretty definitive negative result. There are a few questions, though, that we can get to about are there alternative explanations?
And what's, in your opinion, one possible alternative explanation?
Well, I think one question is, how confident are you that the target was really engaged in the plaque?
No, I think that that's a fantastic question because it's obviously easier to assess Lp(a)2 reduction within the circulation than it is within the plaque itself. And we know based on the dose ranging studies that were conducted before phase 3 that in the circulation at least, there tends to be fairly uniform response in terms of Lp(a)2 activity reduction, around about 66%. And there were also some studies that suggested that Lp(a)2 is reduced to a similar extent within the plaque itself, but harder to quantify. So I think the question of plaque penetration is certainly one that remains open.
I think one of the principles of translational medicine that's sort of evolving now is that the coin of the realm is demonstrating target engagement at the site of activity that's intended. But we know for a lot of therapies that deal with atherosclerosis, that's going to be very hard to do. So I'm not saying it was a mistake. It was just a calculated risk and a gamble. Is that a fair statement?
Well, I think that you're hitting on one of the key issues that I think that this whole field of anti-inflammatory therapeutics is exciting. I mean, the idea of targeting a whole new axis and not just evaluate another antiplatelet or antithrombotic, really trying to delve into a whole new axis of therapy is terrific. But how best to evaluate those drugs before you launch into phase 3 is challenging. And I think you're hitting on the point of, what do we consider to be a reliable surrogate endpoint? Is it the degree of inflammation in the plaque? Should we be relying on technologies like FDG PET enhancement within atherosclerotic plaques or surrogate biomarkers? It's challenging to know the translatability of those into clinical events.
So, I mean, you probably already know about me, but I don't believe there are any valid surrogates. I think they're biomarkers and they will be associated with disease and may even go the same direction as an effective treatment, but reliably predicting the next treatment in a different class, it's a crapshoot. You're taking a gamble. I just think in this case it was an extreme gamble because of all the factors that had to be assumed to be the case that weren't proven, and a company like GSK can do that. I would guess the combination of these two trials was over $600 million, just as an example. So you start thinking about how that money might have been used otherwise. And that gets to one other question I love to ask. How long was the case report for?
It was several hundred pages long.
So in terms of policy of cardiovascular drug development, we just had a meeting and the title of it was, is cardiovascular drug development dead or is it just hibernating? Mm-hmm. And I think the main conclusion was you can't, since it's going to be very hard to get reliable biomarkers that are highly predictive, we've got to take the risk and the gamble, and the only way to make the cost in the range where companies can afford to do it is going to be to collect less data on more patients. And that's a real change. It's sort of going back to the past, the way we used to do things. For whatever reasons, things have gotten sort of extravagant and expensive, maybe unnecessarily so.
This is a crucial point, and Michelle, so you're the time for a final response and a final statement for.
Well, I'd say I appreciate the points that Rob has raised, and I think that there is a lot of considerations that are being given to the best way to make clinical trials more fiscally reasonable. But I think also just the collection of data, I mean, we are able to use a lot of that data to answer a lot of other unanswered questions. So, I would say that the wealth of data we've collected within SOLID will be put to good use. And I think we'll go on to answer a lot of other important questions. So in terms of, what we've learned from the SOLID experience, I think that it appears that direct inhibition of Lp(a)2 with Darapladib does not reduce the risk of cardiovascular events. However, I don't think that this closes the door on anti-inflammatory therapeutics by any means, or argue against the inflammatory hypothesis. I think that that realm is still wide open, and that there are a lot of interesting therapeutics that are being developed in that domain.
I sincerely hope so for the future of research in cardiovascular medicine. And great discussion. Thank you very much.
Thank you.